Greetings, parasite ecologists. Now we come to a really exciting part of our video series where we leave behind all of our familiar metazoan parasites, all of the multicellular parasites, all of the parasites that belong to the kingdom Animalia, and we venture into the protozoa. All of life on Earth evolved from a single common ancestor. There are 3 domains of life: the bacteria, the archaea, and the eukarya. We've been entirely in the eukarya, and that's where we're going to stay as we discuss the protozoa. But I wanted to use this phylogeny to zoom out a bit and situate us in the broader tree of life. If you've taken intro biology, you know that the bacteria lack nuclei, the archaea are extremophiles, and then the eukarya encompass all organisms with nuclei in their cells, including both the protozoa and the metazoa. The metazoa are all the multicellular organisms, including all of the parasites that we've talked about so far in this video series, which are all in the kingdom Animalia. But we're about to step into the protozoa, which are still eukaryotes, but they're different from the metazoa. Now, protozoa is not a strictly correct taxonomic definition because the protozoan group is polyphyletic. You can see that here within the eukarya. Some of the groups that we identify as protozoa, including diplomonads, trichomonads, and flagellates, are mixed in with some other groups that are metazoa. Let's see what that looks like. This is just a different way of looking at the tree of life, all life, except here we've flattened out the phylogenetic tree. You can see that the bacteria and the archaea are close to the root, close to our common ancestor. Then we verge into the eukarya. Everything that you see highlighted in red is a protozoan, and you can see that this is a polyphyletic group because there are lots of non-protozoan clades mixed in here, including the plasmodial slime molds, the cellular slime molds, red algae, green algae, plants, fungi. When a group doesn't contain all of the descendants of a common ancestor, we call it polyphyletic. So protozoa is really just a term of convenience rather than a strictly correct taxonomic term. We define the protozoa, which is synonymous with the term protist, as a polyphyletic group of small, heterotrophic eukaryotes, usually single-celled, although many have more than one nucleus. So remember, this is a polyphyletic group, just a convenient term, not a strictly correct taxonomic term. These small eukaryotes are heterotrophic, meaning that they ingest other matter as their food. They're not photosynthetic, and they are typically single-celled. The first person to ever see a protozoan was this dude, Antony von Leeuwenhoek, who lived in the 17th century. He invented the microscope. And here's what his original microscope looked like. He was interested in looking closer at lots of common substances. One of the first substances he examined was his own semen. He was the first person to see living human sperm. But he also examined his own feces. This is how the microscope works. You put your sample onto the lens and you hold the lens up to a bright light and the other side up to your eye. It's obviously very crude. You could go on Amazon, spend $12, and get a much better microscope than Leeuwenhoek got when he made the world's first microscope. But he was still able to see some stuff. Some of the creatures that he observed were these guys. These are Giardia. Beautiful little protozoa that cause a condition called giardiasis. You might have had it yourself if you've ever drunk water from a stream while hiking. Giardia is one of the most common protozoa that people encounter, and it causes very uncomfortable and painful bouts of diarrhea that can sometimes be difficult to treat. Here's what Giardia looks like on the intestinal lining of a vertebrate host. These are lots of little Giardia trophozoites feeding away on the intestinal epithelium. As we'll learn when we get to the Giardia section, this can result in pretty extreme pathology for the host. Poor von Leeuwenhoek. There are 3 different ways in which protozoa can move around their environment, whether that be the actual environment outside or the environment inside a host's body. They might use cilia. They can use pseudopods, which are extensions of the cell membrane that are basically like little feet. Or they can use flagella. Their reproductive strategies are really diverse, but let me mention a couple of things that they do. They can be either asexual or sexual, and in some species, they alternate between sexual and asexual stages in response to the environmental conditions that they're facing. For example, when things are good, asexual reproduction is a really good way to go because the genotype that you have right now is working well. Sexual reproduction is often undertaken in response to environmental stress, because by recombining your genes with the genes of a partner, you can ensure that your offspring all have a variety of different strategies for surviving in this changing world. For protozoa, asexual reproduction can involve binary fission or multiple fission. You should remember binary fission from introductory biology. It's basically just where one cell splits into two. In multiple fission, the process you get is called schizogony for parasitic protozoa when it produces the same life cycle stage, and it's called sporogony when it produces a different life cycle stage. Don't worry too much about that right now. We'll talk more about schizogony and sporogony when we get to the malaria parasites. A lot of protozoa, particularly parasitic protozoa, undergo this process called encystment, where they secrete a resistant covering that provides protection against unfavorable conditions. Sometimes when they're inside their cyst, their structures just kind of de-differentiate or degenerate, and nuclear divisions can sometimes give rise to more than one nucleus in this stage. And that's just because these guys are laying in wait, waiting for their opportunity to infect another host. The cyst provides them with protection. They don't need too much structure in order to get along in that state. This cyst stage is really important in epidemiology. For example, one protozoan that we'll talk about, Entamoeba histolytica, is frequently spread by people who are totally non-symptomatic, but they're carriers of cysts. They poop out those cysts, and the cysts get into other people, some of whom might not be so asymptomatic. I want to take a moment to emphasize that even though these protozoa are just lonely single cells, even though they're a lot simpler than some of the parasites that we've seen before, they are still quite beautiful and capable of doing some pretty wild things. Here is a list of some of the groups of parasitic protozoa and example species in each group. All the taxa that are not taxonomically proper are shown in quotation marks here. You'll see that protozoa is in quotation marks because it's a polyphyletic group. It's not a proper taxonomic definition. You'll also see that there are a bunch of other groups of protozoa that are similarly grouped by their morphological, ecological, or biological similarity, and not necessarily by their taxonomic affinities. This includes the amoebae, the flagellates, and the hemoflagellates, which, from their names, you can probably guess, are united by their similar modes of living, either movement through the environment or the places where they live, with the hemoflagellates, for example, being flagellates that live in blood. We do have one proper taxonomic name that we'll talk about, the phylum Apicomplexa. We won't discuss all of the species that are listed in that phylum, but we're going to talk about two representatives, Toxoplasma gondii and Plasmodium, the parasite that causes malaria. So that's what we're going to cover as we move through the protozoa. I'll see you in the next video.